Who is Don Peter Häder?
In the beginning, I was interested in cyanobacteria. Interesting way of putting it.
How did you become interested in the field of gravitational biology and space biology?
We did not solve that riddle, so we looked into the organism and found out that this is fairly interesting. Nobody really knows the mechanism for graviorientation in humans. The centrifuge on board of the shuttle. So we put the organisms inside.
What role does gravity play in the evolution of different life forms on Earth?
And all the astronauts feel that they have a reduction in muscle volume. The parabolic flight gave us medication. Is that a plant or is it an animal? These cells, Euglena, does not have sexuality. Again, we don't know how they do
that. Hello and welcome to Rocket Biologist, the show where we meet experts in medicine and biology. I'm your host, Dr. Adil Nasir. My cosmic comrades, welcome back. In case if you have missed our previous episode, catch up via the link in the description to fully connect with today's conversation. Let's dive straight in. The word taxis basically means that locomotion of one organism from one point to another under the influence of a stimuli that can be gravity, magnetic field, and so on and so forth. The idea is that for the Gravitaxis, that what is known that in nature, as you already showed that mentioned that earlier, that when you go out and you see on a puddle and there is a Euglena just sitting on the surface, which means that they are showing negative Gravitaxis. They are going away from the source of gravity. Right. And when you culture them in a lab in the domesticated conditions, they show positive Gravitaxis in the initial days of culturing, and in later as the culture age, the orientation of Euglena cells is like changed, so it's from positive to negative. The one of the criticisms that that comes to your research or your hypothesis that you have, that the negative Gravitaxis, what you have always shown through different important work of your life, that it's a physiological mechanism. But a lot of authors kind of challenge you on that regard. They say that the shape of the Euglena cell in such a way that this has a bottom heavy, and that's why they are moving. They're showing that kind of behavior, but so it's not physiological behavior, it's pure physical behavior. Care to respond to that critique?
Okay. These were basically the physicists who built the hardware for the rocket flights, which we did from Kiruna. There are several indications that this cannot be true. First, when you look at the cell under the microscope, I don't see any heavy material at the bottom end as compared to the front end, in contrast to some ciliates where you do see a gradient in dense material. Second, as you mentioned, the fact that they switch from positive to negative Gravitaxis without any obvious change in their morphology seems to indicate that there is some physiological aspect involved. Third, when you use inhibitors, which can be some metals like vanadate, you can change the direction of movement, and you can use other inhibitors, and that's point 4, which completely wipe out Gravitaxis. So the cells go random in various directions and no longer know where is up or down. Okay, I think that was the initial indications that this is a physiological, not a physical mechanism. Then we started looking into what could that be. There are certain proteins in all kinds of organisms, including humans, and these proteins sit in the membrane and they allow the entrance of certain ions. This is used in the mechanism of vision, also in humans, in hearing, in chemical gradients, in temperature gradients, or in pain perception in humans, for instance. These are proteins which, under the external stimulus of whatever I just mentioned, can open and close and allow the influx of certain ions. In this case, in Euglena, happens to be calcium. So if you take all the calcium out of the medium, they will not respond to gravity. Okay, so when you have to trigger these proteins by minute forces of gravity. Imagine the cell is less than 100 micrometers long. So you can calculate the force. We know the cell is a bit heavier than the surrounding medium, than water. It has a medium which is 1.04 grams per liter. And when you calculate that, then you come to piconewton, which is an incredibly small force. How can they measure that? Here comes what I said before, the helical movement when they swim like on a helix. Um, let's assume they are swimming horizontally, and we know, oh no, we assume that these proteins, these gravi receptors, are located at one side of the body and only in the front side underneath the flagellum. So when they rotate, the proteins will be either down or up, and they take that difference in the signal to open or close these proteins. We can show that, and we did that on rocket flights using fluorescent dyes, which indicate the presence and entrance of calcium into the cells. And that correlates when you're at microgravity, there's no influx of calcium into the cells. And when they go back into 1G or increased gravity, you see, oh, there is an increase in the fluorescence, in the calcium fluorescence inside the cells. Okay, so that was the first indication that there is a physiological mechanism, and that was not the end of it, but rather there is a fairly complicated biochemical cascade inside the cells, which allows you, well, eventually triggers a reorientation of the flagella, which reorients the movement of the cells. So the calcium has to be bound somewhere. Well, one of the typical receptors for calcium is Calmodulin. In fact, things were becoming a bit more complicated. We had a Russian biologist here who was trained in molecular biology, and he went deep down into the genetics of Euglena, and that has continued ever since. And my current co-workers keep telling me they look into thousands, literally 10,000s of genes to see what might be involved in the next steps. So when we look into Calmodulin, there were 4 different Calmodulins in Euglena. Humans have even more, and they were very specialized. Only one was capable of triggering the gravitational cascade. The others were not interested in it. So there's a mechanism by which you wipe out genetically a certain molecule. So you kill the gene for the calmodulin, and then look at the culture which comes out if you kill out calmodulin number 1, nothing happens. They still orient number 2, number 3. But when you wipe out number 4, then the cells cannot orient anymore. So this was the one calmodulin which was important for the orientation mechanism. But that was not the end of it. Then there were next steps, and I don't want to go into all the details, but it involves cyclo-AMP. ATP is the energy form in most of the cells, also in the human body, and cyclo-AMP, adenosine monophosphate. That was interesting because our Japanese friends from Okazaki had shown that this cyclic AMP is also involved in the photo orientation of the cells. Okay, so this is the point where the two responses get together. So we have to find out what cyclic AMP does. So when we get rid of that molecule within the cell, again, no Gravitaxis. And the next step is what does the cyclic AMP do? Well, that's kind of interesting. It modulates a protein. Well, there are tons of over 100 proteins in the flagellum where eventually you would see the reorientation mechanism. When you look under the microscope, under certain stimulus, either gravity or light, then it whips out the flagellum, which usually goes back parallel to the cell body. It whips out to the side, and by that it does a reorientation, a steering mechanism. So one of the proteins has to be inside the flagellum to facilitate that reorientation. And in the end, we found out which protein that is. So again, when you block this gene for that protein, again, there's no reorientation with respect to gravity. So this is a fairly complicated mechanism, and it seems to be even more complicated. Question is the mechanism the same for positive and negative Gravitaxis? What is the switch to go from one to the other? What are other stimuli mechanisms which modulate the system? Usually you don't have one receptor, one response, but you have to have a modulation, and that depends on a number of different other factors. Again, in human beings, when you look at the photoreceptor, you don't have one photoreceptor in your eye, and that tells you, well, there's light, but you have several photoreceptors, and they have to work over a long range, logarithmic range. Densities in starlight is different than in full sunshine. So you have to adjust the sensitivity over several log units, like 7 logs. So that is interesting to see what the other mechanisms are which modulate the system, and we don't know that in Euglena yet. As I said, our current co-workers at the university lab continue doing that, and things become more and more complicated. And that's a truth in science. Things are always more complicated than you think they are.
Well, that's the reality we are living with. Keep on working. That's it never ends. The curiosity keeps you moving. So, Professor, just you summarize quite well about the it's a complex mechanism of signal transduction chain that uh, takes that through which Euglena can really perform the gravitaxis, the mechanism of negative gravitaxis, and fairly, this is still under study. You still want to find more. So, do you have any what would be your? Do you still wish to investigate, if you have to prioritize something that you still wish to investigate, and you think that you would like to conduct an experiment if you get the chance yourself? What would that be? What's still missing?
Yeah.
The missing link.
Well, it took us several decades to find out the basic mechanism for reorientation in Euglena. So somewhere, not a scientist in the population could ask you, well, that's interesting, but uh, who cares? Why should I want to know how Euglena orients within the water column? Well, I agree. This is a mechanism, a pet organism, which hence gives you a hand to study the mechanism. And as I said, we are far from understanding everything in there, and we still do active research both in the lab and in space to get further answers. But as I mentioned before, it will be interesting to understand the mechanism, for instance, in humans. What is the mechanism of reorientation? When you think of what I said before, I still think there are also these proteins involved, which, well, there are more than 24 of these proteins, which we know by now. There may be many more. Um, I'm not sure if calcium is involved in light perception, for instance, in the human eye. There are other ions involved. It would be so I don't know what it is, but the first step would be to study that. And one of the places where you could do it is at the DLR in Cologne. And I keep uh, itching my colleagues there to do the experiment. They have a centrifuge with a built-on microscope, and they can do fluorescent studies. So what I would like to do is use a one of the novel calcium fluorescent dyes to see if or not human cells. You can take isolated human cells which respond to gravity to see if there is a calcium flux during gravity stimulation. That would be one thing I would like to know. And I think if you were capable of proving that and showing that this is the first step, or could be any other ion maybe, that would be a very big challenge and would be worth a Nature paper. So I keep telling them, well, why don't you do this experiment?
Well, I hope that they listen to you and conduct that study. That would be fascinating to see how this study can, teach us a lot in the time of space age when human want to colonize other planets. I think that would be a
Uh,
relevant study. So you did mention earlier that Euglena has this capability to really sense partial gravity. Do you think that that capability of Euglena makes it suitable for, for if one day, I mean, I'm not suggesting we should terraform another planet that, which has a
yeah,
partial
there
gravity,
were
and
these
let's
ideas
suppose that it also has,
terraforming Mars. But well, when you
Mhm.
think about the details, what they thought doing was using cyanobacteria and put them into the atmosphere. But then, Mars has only 1% atmospheric density. There's hardly any oxygen um, in the atmosphere, and when you use, if you consider using cyanobacteria, first of all, they have to stay in the atmosphere, which might be difficult. Second, you have to have uh, water, which is also a problem on that planet in order to split the water and produce oxygen. So that has taken a few billion years on our planet. So this will not be done in a short time on Mars. So I don't see any possibility of doing that in the short range on a planet like this. On Mars, other options where people concentrate right now are the moons on Jupiter and Saturn, which may have uh, watery um, ecosystems under the solid ice surface. So maybe there's some possibility. Well, they do have enough heat in the inside the moon due to gravitation host planet like on Jupiter, which could be sufficient to produce liquid water, and that could be in this all mere speculation. Nobody has ever had a chance of looking into that. That some life forms might have developed or might develop there. This is, but as I said, dreamland and not backed up by any scientific knowledge.
Why do you think, in the first place, that Euglena acquired that kind of ability to sense different magnitudes of gravity?
Well, as I said, all almost all organisms, except very small ones like viruses or very small bacteria, do have that capability, and I think that is built in all living cells in order to, and which is very helpful in order to find a place in their environment which is suitable for survival, giving them light, giving them chemicals, organic material which they can metabolize, escaping detrimental effects like, for instance, high UV radiation. Imagine at that time when these cells, when the first cyanobacteria, for instance, developed, there was no oxygen in the atmosphere. Consequently, there was no ozone in the stratosphere. And that means they not only got UVA, UVB, but also UVC all the way down to 200 nanometers wavelength. So you have to escape all these detrimental effects, and the more sensors you have to avoid dangerous um, situations and to take advantage of good environmental conditions, the better for you. So using light, using gravity, chemical sensors, maybe even magnetic field, which Euglena does not have, but other organisms do have, like some bacteria, which orient with respect to the magnetic field. That would be advantageous to for survival and reproduction.
Okay, so basically, Euglena is a water inhibiting organism. So the impact of gravity, when you talk about the terrestrial life and the life in the water, it changes a bit, right? So can that be a reason that it has adapted to different magnitudes, that is still sensing the using this although the gravity vector remained constant throughout the evolution of Earth? Do you think it could be something related to that, or I'm just speculating completely wrong?
Well, what we know is that these signals don't operate independently. They do interact depending on what is stronger or more important. We think it would be a possibility for the organisms to uh, better adapt. So if we have changing light conditions on Earth, like our increasing temperatures right now, thermal sensing I did not mention before, which is also a very important factor, of course, could change the responses of these organisms. But any change in their evolution or in their morphology, in their physiology, would take a long time, especially if we talk again about Euglena. Since you don't have any sex, they can't exchange genetic information. So it will take, well, if you think about Darwin, it's a process which will take a long time until we will see any change in their behavior within the habitat, within the ecosystem.
Does you studying gravity sensing in Euglena gracilis teaches us anything about human and bacteria gravity sensing mechanism?
Well, this is a model system, and we were glad and lucky to be able to unfold this and try to understand the basic mechanism, maybe not all the details. But that does not help you with all the other organisms. As I said, well, we don't know anything about humans or any other animals. We know a bit about the mechanisms in plants, uh, where it's sedimentation of heavy um, organelles within the cell, like starch bodies. But we are still working on it to understand what the subsequent steps are. We know when you look at a cell in a gravity field in a microscope, then you see that these starch bodies fall down, press on the lower membrane. What they do there, if they modulate the influx of ions, which we know there are, we don't know whether well, we also know that it modulates, for instance, the growth stimulating hormones, but in what way they do that, we don't know. In fact, when you have one organism, that doesn't help you with all the other ones, and there are many. So, in order to understand what happens in like ciliates or in ferns or mosses, which have been studied to a certain extent, or in some of the bacteria, where it has been also studied in larger bacteria. There's a group in the United States which does that. That is a totally different story. So life and biology is, as I said, much more complicated than initially you think it is. So the biologists will not be out of work, but rather there are a lot more new questions and with new technologies, like also in space biology, we will be able to understand a bit more in certain organisms, but certainly we are interested in humans and do all kinds of experiments, both on Earth, on centrifuges. Like in Cologne, they have a large centrifuge where they can accelerate people to 4G, or rest down, um, head, head rest down research where people are forced to lie down to mimic the different distribution of blood in the cell body, all the way up to the space experiments on the ISS, or maybe even one day on Mars. But that will take a little more time until we will do that, and I'm not so sure if that is really taking us to very new ideas. Maybe it's a challenge.
Okay, so do you think, or what the studies shows that what should be the size of organism that it can, minimum size that it can sense gravity? For example, with a lot of bacteria, the gravity sensing mechanism is not really clearly defined. If they're really sensing gravity or it's an indirect effect, but in microgravity conditions, a lot of bacteria behave differently. Sometimes their replication rate is reduced. They are growing much faster. In some cases, it doesn't happen. So, what is happening when they are exposed to micro? Do they have a gravity sensing mechanism?
Well, you have to consider the forces which are produced by the difference in density within the cell and outside. So that depends on the volume of the cell. So let's assume the difference is like 4%, like in euglena or other cells. Ciliates are on the same level, like 6%. Then you can calculate uh, at a given volume what force you can produce in terms of piconewton. Well, if you then calculate what you need to open or close an ion channel in the membrane, if we assume that this is the mechanism, what happens, then you can define a certain cell size which would be just capable of sensing or not sensing. If it's not this type of mechanism, but maybe a physical mechanism, like one end of the cell is heavier than the other, then this may be a totally different story. Or in the case of some magnetic bacteria, which use iron compounds which work like a magnet inside the cell, which causes them to go parallel to the magnetic field lines of the Earth. That would is a mechanism. We know that some of the bacteria do that. And then the next question is, well, this happens on Earth. How about Mars, which does not have a magnetic field? How would they orient there if given an environment with water, the correct temperature range, and other factors, then they would not have the magnetic orientation. So that's a kind of an open question.
All
So
right.
so that all depends on what the other factors are, like light sensing. We know some of the bacteria, like halobacteria, have a capability of measuring light. This is not a phototaxis, not a direct movement with respect to the light direction, but a random walk mechanism when they go up in a light gradient and feel it's getting too much, they will return. And so they go back and forth, and depending on whether they go more forth than back, will bring them closer to the light source and the other way around. So you have to consider all the other sensors as well as gravity, which is certainly one important thing. But what happens if you are on a smaller planet or on a moon, which has a very low gravitational field? How would organisms respond to those conditions? And we sort of mimic that by looking at that in space under such conditions.
All right. So basically, you did study uh, space biology, like the applications. You performed a lot of projects. One of the projects that we use Euclina for the biological life support system and for space studies. Would you like to tell us a little? Because I would like to have another complete episode on that, just to give the teaser now. So if you can tell us quickly.
Okay. Just imagine you want to go to Mars or any other extended space research. Then you have to enough food. So that's a lot of bulk and volume. But then you need oxygen, of course. So if you calculate how much oxygen one astronaut needs per day or per hour, whatever, you can calculate doing an extended space mission, let's say half a year or a year, going to Mars, depending on the position where the planet is right now with respect to the Earth orbit. You can calculate how much oxygen you have to take. In addition, you have to get rid of the CO2, which we do by respiration and exhale. So you have to get rid of that CO2. Do you blow it out of your spacecraft, or what do you do with it? There are absorption systems which had been used even on the Apollo flights when they went to the moon. Um, Water is not a problem because that is recycled, which doesn't sound too nice, but it's being used on the ISS and other space missions. So oxygen and CO2 is a problem. So now we know that, for instance, Euglena or many other organisms do photosynthesis, and we know that they produce oxygen. We can calculate how much euglena you need to keep in or produce enough oxygen to keep one astronaut alive, provided you have enough light. So when we do that in the solar system, close to Earth or Mars or Venus, there is certainly enough light. We need a window which is about 1 square meter filled in a cuvette filled with euglena to produce enough oxygen. And the other thing is, euglena during photosynthesis takes up CO2 to produce organic material. So we can solve these two problems by having photosynthetic organisms. There are several groups on Earth in several countries, like in the Netherlands and Belgium and the States, which have developed various mechanisms and equipment, also in Russia, to use microorganisms, photosynthetic organisms, to produce sufficient amount of oxygen and take up the exhaled CO2, which would be also toxic for the astronauts. So this is an approach which we have tried to study in a number of experiments on photon satellites, which we have been flying usually about 8 or 10 days to see if it's possible that a culture of these microorganisms is capable of producing oxygen and taking up the CO2.
Wonderful. Well, we would definitely continue on that subject in the next episode, hopefully. And this is like hypothetical questions, and I would definitely would like to have your, wise opinion on that. So, if you're comparing, an organism that's going to adapt to microgravity conditions as we are heading to the stars, which organism has more chances to adapt faster to the partial gravity environment? It's bacteria, unicellular eukaryotic organisms, plants, human. What's your pick?
Well, when we talk about the timescales involved, I have serious doubts that any of the terrestrial organisms would be capable of adapting to gravity. We could, make maybe able to work with altered life conditions like different gravity conditions. The astronauts are showing that it is possible. They have some consequences like on the blood flow, blood pressure, muscular volume, physiological processes. Euglena can, for instance, adapt to microgravity. They don't die when you keep them in a prolonged um, situation of reduced gravity. But adapting in genetical terms, I would say, would take a very long time, which is beyond the time scale of our lifetime and work time, which we have for them. So that does not really work. Well, even if you go to Moon, which has 1/9 of the gravity of Earth, according to its size, we still feel the gravity, but you can jump much higher and run faster. But then you would have to kind of adapt, which is not a genetical adaptation, but a rather a physiological adaptation, which you do on your body. So if they ever there are concrete plans to do that. Build a station on the moon. You would have to consider that and take into account that you have different conditions for your life in that environment.
Wonderful. So, last question, and then we'll wrap it up. So, what's your advice for non traditional spacefaring countries to be part of this space journey?
One of the basic trigger of science is the desire for knowledge. When you imagine people about 200 years ago who went to Africa, they knew this is dangerous and has different um, difficulties in the environment. but still they went, even at the loss of their lives. And the same was true when people went to Antarctica, like Scott and others. Well, many people have died in the rush conditions in Antarctica because we didn't have the technology or the protection mechanisms. But still, people do that because they're curious. And I think curiosity is the main point in science. We do that because we want to know how things work. And as I said before, each time you do that, you find out, well, things are much more complicated than you think originally. So when you ask a question, and this is also true for modern space technology, you find 10 new questions which you have to solve, and that will continue. And I think that's a good point. Investing money, investing time and work to further our knowledge. And obviously, that has helped in the history of mankind when we started to looking more and more into details. When you think about medicine, about food, in even the ethics of the collaboration between people, it doesn't always work. We still have wars, and we still have bad conditions in terms of medicine and diseases. We do have problems in the environment, which we actively and involuntarily change. But I think that's the only way to, over the very long time, to adjust and better the life for people on Earth, and well, maybe, I don't know, in the future in space and other environments.
Thank you very much. That was lovely talking to you, Professor, and I hope that we can continue our conversation in the next episode. Once again, thank you very much, sir. It was an honor to have you.
Well, thank you very much for this interesting discussion and exchange of ideas. So I'm looking forward to a continuation of that. So thank you again.
Goodbye, everyone. See you in the next episode. My cosmic comrades, we will continue our intergalactic conversation in the next episode. Until then, stay curious and stay connected as we are just beginning our odyssey into the heart of the universe.